Okay, thank you to the Foundation for this great opportunity for me to present my research, which is done in Dr. Ryan Potts' lab at St. Jude's Research Children's Hospital. So this work is a highly collaborative effort from many different labs in many different institutes, and most of them are funded by the FPWR Foundation. And as you can see that we were fortunate enough to be accepted for publication in JCI Insight recently. So what we're interested in is understanding the role of MAGEL2 in Prader-Willi syndrome. MAGEL2 is interesting to us because mutation of the protein alone is causal for the related Shaff-Yang syndrome, and there's highly overlap between the two pathologies. Well, we don't have a lot of understanding in regards to MAGEL2's role in Prader-Willi. What we do know is its physiological role using various cancer models. In our previous publication, we have shown that MAGEL2 essentially regulates protein recycling and protein trafficking on the endosomes, and how this happens is that MAGEL2 activates a protein complex called WASH, and this directs membrane proteins either back to the plasma membrane or to the transgolgi network from the endosome. And in doing so, these protein cargoes are prevented from undergoing lysosomal degradation, and their stability is maintained within the cell. At the molecular level, essentially, MAGEL2 functions as an activator of the WASH complex. So based on that, some of the questions we're asking is this pathway defective in the Prader-Willi background, given the loss of MAGEL2 expression? And if so, is this pathway affected in specific tissues of relevance, such as the hypothalamus, as well as a cell type of relevance, such as neurons? And if that is true indeed, what are some of the key cargo proteins that may be affected, i.e. their abundance being lost in these relevant tissues, and how does that translate to some of the pathology behind PWS? So before we begin answering that question, the first thing we wanted to do was generate a MAGEL2 antibody. As most of you know, that there is really no reliable commercial antibody out there for us to purchase. So what we did was that we generated a polyclonal antibody against a mouse peptide, the C-terminal domain, specifically because of its highly conservation with the human protein. And to test the validity of our antibody, we looked at wild type and paternal MAGEL2 knockout mouse, and we collected brain tissues and we stained them using immunohistochemistry and corroborated our results with what's been published in the Allen Brain Atlas in terms of their in situ hybridization data. And from what we see is that there's a high expression of the MAGEL2 transcript based on our antibody in the hypothalamus and the amygdala of the brain. Specifically, we see high immunoreactivity within the cytoplasm of neurons, where it's not found in glia or astrocytes. And using this antibody, we're able to robustly validate the loss of MAGEL2 expression in the paternal knockout animal in hypothalamic tissue collected at multiple time points. The next question we wanted to ask is, what are the MAGEL2 regulated processes that may contribute to PWS pathology? To do so, we did a collaboration with Jim Maresco's group in the Scripps Institute, and what they did for us was they performed quantitative mass spec analysis on hypothalamic tissue we collected of wild type and MAGEL2 knockout animals. And so from there, we identified 2,800 distinct proteins. However, only 52 proteins were actually altered in terms of their abundance. Specifically, 40 proteins were downregulated in the knockout animals. So from there on, we performed gene ontology analysis to look at which pathways are indeed affected. And nearly half of these downregulated proteins were components of the dense core granule or secretory granule pathway that's involved in the processing and releasing of neuropeptides. So just a brief overview of secretory granules. So they are essentially hallmarks of neuroendocrine tissues and cells such as the hypothalamus. They essentially function as exocytic organelles for the processing and secretion of hormones and neuropeptides. So within the regulated pathway, here on the right, as you can see, hormones and neuropeptides are normally synthesized as large inactive precursors in the ER. Then they're packaged, processed to become active bioactive peptides in the secretory granules. So once they become active, they're stored within the neurons and then released upon stimulation. So for these secretory granules to occur, what happens is that granin proteins such as chromogranin A and chromogranin B, they essentially coalesce these prohormones into the secretory granules. And within the secretory granules, there are enzymes such as PCSK1, PCSK2, and CPE, and their function is to process these prohormones into their bioactive cleaved forms. And based on our data, we see that various enzymes as well as these granin proteins are downregulated in the knockout animal. As well, at the same time, we see a downregulation of various neuropeptides such as oxytocin, AVP, and somatostatin. To confirm our mass spec finding, we performed Western blot analysis on wild type and knockout animals at different age time points, and we see that across the board there's a decrease in the level of the secretory components in the hypothalamic tissue. From there on, we want to extend our findings into a more human PWS relevant disease model. So this collaboration was done with the Tchaikovsky group at the Broad Institute. And what they did was that they generated human-induced pluripotent stem cells with a paternal type 1 PWS deletion using CRISPR. And once we got those cells, we're able to differentiate them into neurons by this induction of a neurogenin 2 overexpression. And with these, this method of differentiation into neurons, these neurons at day 7, they show electroconductivity and they're excitable. But for our assay, we chose to extend their maturation for another 7 days for day 14 as the final time point. And here we see that using quantitative Western blot analysis, we see that the PWS-induced neurons or eye neurons shows reduced level of secretory granule components compared to the control eye neurons. And using a time course assay, we see that this reduction in protein expression is consistent throughout the entirety of the differentiation. Moreover, we see that some of the key neuronal marker proteins such as MAP2 and TUB3 are the same between the eye neurons and between the two genetically distinct eye neurons. We see suggesting that the differentiation rate or maturation rate of the PWS and the control eye neurons are the same. So to build on top of that, we want to extend this to a more patient-related model. So this work's done in collaboration with Dr. Larry Rader's group at University of Tennessee. And what he does is that he collects dental pulp stem cells from PWS patients, and then he generates them into neurons using a chemically derived methods. So from there, we analyze some of the PWS patient-derived neurons. We see that the expression level of the secretory granule proteins are consistently less compared to the age-matched control. And at the same time, we're able to examine some neurons generated from Shaft-Yang patients, and we also see this defect carried as well, suggesting that MAGEL2 indeed is a major player within the PWS locus that contribute to the stability of these secretory granule proteins. So the next question we want to ask is, how are these proteins lost within this system in the absence of MAGEL2? So is it either happening at the transcript level or at the protein level? So to start, we looked at the transcript level of all of these secretory granule proteins by QRT-PCR. And in the mouse, and as well as this, what's shown here, which is a human eye neurons, we see there's no difference between the transcript level, suggesting that the change is likely occurring at the post-transcriptional level. So from there on, we want to see whether the proteins are lost through lysosomal or proteasomal degradation. So for the eye neuron cultures, we treated them either with a proteasome inhibitor or the lysosome inhibitor. And the baflomycin A1 or BAF here is a lysosome inhibitor. We see that with that treatment, we're able to restore the expression level of all these different secretory granule proteins, suggesting that indeed, in the absence of MAGEL2, they are lost through lysosomal degradation. So based on that piece of data and our previous knowledge of MAGEL2 is upstream of what the WASH complex regulating protein recycling and trafficking on the endosomes, and in the absence of that, these cargo proteins are not trafficked properly or recycled properly, but indeed they're actually undergoing lysosomal degradation, affecting their protein stability. we postulate that if we were to knock out components of the WASH pathway, which is downstream of MAGEL2, we would be able to recapitulate the same finding. So we were able to collect some of WASH knockout animal hypothalamic tissue. So these animals are Nestin-Cree conditional knockout. And from there, Western blot shows that indeed PCSK1 and PCSK2 levels are reduced dramatically compared to the control wild-type animals. And at the same time, we performed some immunofluorescence analysis in the PWS eye neurons and the control eye neurons. So here in the control one, you can see that the granulin protein chromogranin B co-localizes with the protein WASH, but this co-localization is only seen at the soma of the neurons, but not within the neurites of the processes of neurons. So this suggests that the co-localization suggests that indeed this is where the recycling is occurring within the cells, which is in the soma, but not in the neurites. So all of those data are very convincing of there's a disturbance in the protein stability of these secretory granule proteins upon the loss of MAGEL2. So we want to see what the downstream effect is, which really to us first means the number of secretory granules or their biogenesis. So to do this, we looked at a combination of models. So we have our paternal knockout mice, we have our human eye neurons with PWS deletion. We also have our DPSC-derived neurons collected from PWS patients. So using a combination of electron microscopy and immunofluorescence, we were able to consistently quantify a decrease in the number of mature secretory granules within all three systems. And the downstream effect of that is the next thing to look at the level of neuropeptide processing or and release. So to do, and as most of you already know, that the hormone deficiency is a universal feature of PWS. So within the mouse model, we looked at 5 different bioactive forms of these neuropeptides as they came up in our initial mass spec screen. And looking at the plasma level, the sera of the knockout animals, we see that oxytocin, AVP, somatostatin, growth hormone, and MSH are all significantly less compared to the wild-type counterparts. And moving on to our 2 human models, on the left-hand side, you see this is a human eye neuron culture. We're able to quantify using ELISA the level of oxytocin released into the media at both the basal level as well as with treatment of potassium chloride. And under both conditions, the PWS eye neuron shows reduced level of oxytocin release compared to the control counterparts. And the final piece of data, which is the most interesting of them all, this was done in collaboration with the Columbia University as well and Levo Therapeutics. And what they did was looking at the level of plasma chromogranin B level in PWS patients. So chromogranin B is one of those granulin proteins that coalesces the neuropeptides, and at the same time, they're also released into the plasma along with the bioactive forms of the hormones or neuropeptides. So when they quantified this by ELISA, they saw that the PWS patients showed less or reduced level of chromogranin B within the plasma compared to their counter healthy parts, the siblings, as well as their healthy parents. So what I hope to have shown you with this work, or when you read this paper, is that with the paternal loss of MAGEL2, there is decreased availability of secretory granule proteins within the hypothalamus and neurons. And this is due to the fact that there's insufficient protein trafficking or recycling pathways mediated by WASH on the endosomes. And the downstream effect of that is that there's overall reduced abundance of mature secretory granules within these neuroendocrine tissue. And as a result, there's a global level of reduced processing or of neuropeptides and hormones, and therefore a reduced release of circulating hormones. So as I mentioned previously, this work is a highly collaborative effort done through many labs and many institutes, and they are really great collaborators of ours, and we're highly appreciative of their effort on this project. And I just want to thank FPWR for their continued support and funding, as well as St. Jude Children's Research Hospital for funding my fellowship. So thank you for listening.